So, today I'd like to talk about the stream for visually guided actions. We saw way back a few lectures that from the visual cortex, you get two streams forming. One called the Watt stream that goes underneath the brain to the inferior temporal lobe, and one above the cortex that makes synapses with areas in the parietal lobe. And the one heading up is called the dorsal action or wear stream, and it's mainly concerned with the representation of space for guiding movements. And this stream underneath here we saw was concerned with the perception and recognition of objects. Now there's a variety of movements or representations of space that are coded up here, and the representations of space are to guide different types of movements. There's rapid eye movements called saccades, there's grasps, there's reaches, and there's feeding behavior. We'll look at each of these in turn today. And one's attention is focused when we make a movement onto whatever object we're going to make a movement to. But the movement itself is unconscious. So we don't think about the details of reaching for this mouse. We just say, okay, it's the mouse I want to reach for. The other thing that's different, whereas in the Watt stream, we are conscious of the things that we see. The other thing that's different is that in the Watt stream, we have these perceptual illusions. Okay, so these lines look like they're of a different length, but in fact they're the same length. And they're a property of the visual Watt stream, trying to turn these into objects, and these objects being part of the scene. The Watt stream has to have a very accurate representation of what the length of lines are. So if you're going to throw a dart to here, you can't be misguided by what the Watt stream is telling you it is. So the Watt stream has its own independent guiding of movements, and it is accurate. It's not distorted by the perspective illusion. Now the other thing that's different is they use different frames of reference. We have the ventral stream, which is an object-centered frame. So an apple is relative to a table, an eye is relative to the face, and so forth. Whereas this stream, the dorsal stream, wear stream, is an egocentric frame. And there's a variety of egocentric frames that you could have. One is when you look at things with your eye, you see them with respect to your eye, so it's with respect to your retina. And the retina is things that are with respect to the center of the retina, the fovea. So are they to the left of the fovea? Are they to the right of the fovea, above the fovea, that sort of thing. When you hear with your ears. Those are located in your head. And when you hear a sound to your left, it's to the left of your head that you're talking about. So if you turn your head towards a sound, it's then coming directly towards the head, and both your ears hear it equally. Whereas if you look at where your finger is coded with respect to your body. I can touch these two fingers together, my left and right index fingers with my eyes closed. And so we have this some sort of internal representation of where those fingers are located with respect to me. Now, patients that have lesions in these areas have difficulty making either saccades or grasps or reaches or feeding. Now, much of what we'll be talking about is concentrated about this sulcus called the intraparietal sulcus. And it's located here on the top of the brain. This is the front, this is the back. Vision comes in here. So this is, again, very well located to combine this information. So we have this touch information coming from behind the central sulcus. It has the whole representation of the body here. And all that touch information about where your fingers are or where your toes are is coming this way. The sounds are coming in this way, and it's relative to your head. This was relative to the body. Vision is telling you where things are relative to the eye. Now, they all have to get somehow coordinated. So when things are relative to the eye and when things are relative to the body, you want to reach for it, you've got to get into some sort of common frame. And that's all done along this IPS. Now, one of these areas, called the parietal eye field, located here in yellow, is involved in locations with respect to the eye. So there are objects you intend to look at, like I want to look at these strawberries. So I see them to one side of my visual field, and I make an eye movement towards them, and they're centered in my visual field. And neurons here respond to both visual and auditory stimuli. The other thing you have is a parietal arm field, and there's two locations where that is being coded. And it is, again, represents retinotopic locations that are in the immediate peripersonal space. And there are things that you can reach for. And the parietal arm field is used to direct these movements. So neurons here get visual information and somatosensory information. And when you have lesions here, then you have errors in reaching to things accurately. Another field up here, which sounds similar to the parietal arm field, it's called the parietal grasp field. And it's not where objects, coding, well, not where objects are, but sort of shape information that helps us grasp it. So, for example, when I grasp this strawberry, the shape information, well, I can see that it's round. But what's more important is that I recognize that it's a strawberry, and therefore I should pick it up gently. If I were to pick up a knife, I would pick it up very differently. I'd make sure that I picked it up by its handle and not by its blade. And then we have this an odd-sounding structure called the parietal face field. You might not think that have thought of something like this, but this is actually very important. And it's involved in feeding, something we do every day many times. And it represents space that's very near you, and it's used to guide your head, like turning your head, your mouth, your lips during feeding, or other acts like kissing. And it has visual input and tactile input. From tactile input that comes from the face touch sensors. So we see that we've got all kinds of egocentric regions. The parietal face field codes very near space. Others, like the parietal eye field, code space that's near and far. So, and then finally we have the grasp field, which not only gives information not only from vision but from the what stream, because that then tells us what the object is. Yes.
Again, what the IPS does very similar to what the parietal and temporal occipital cortex does.
No, most of that, meaning the inferior temporal.
So the region we studied last time, that's one of the association areas, and one of the structures is integration.
Oh yes, yeah, PTO. Yeah. Oh yes. No. This part here is part of the PTO. Yes. Yes. Oh no, it does. This is sensory information. The touch information comes from this area here and heads back into this cortex. Yes. Okay. So, in different shapes. So one of these areas, the parietal face field, we'll study in a moment. So neurons here will fire when something touches a particular part of the face. So one neuron will fire when something touches the lip here. Another might fire when something touches the forehead. And it not only fires for something actually touching, but something about to touch. So if it sees something approaching that mouth, so it's visual and tactile. And it doesn't matter from what direction it's heading. So it can figure out whether it's heading, if it's heading from above you or below you. As long as it's heading for that particular spot on your face, that neuron will fire. And it doesn't matter where you look. So that's important because it tells you that that's not coding with respect to the eye, because the eye is moving. It's coding with respect to the face. And then it's not activated if something touches the brow. So that neuron that we were looking at a moment ago would be silent. But there's another neuron that we could locate that would be active for this particular spot on the face. Okay. Different areas are involved in directing saccadic eye movements. And we'll see in a moment that what areas up in the parietal lobe are involved. But we saw in the first lecture, or the second, that information from the eye goes directly to the superior colliculus. That information comes from the peripheral part of the eye, those big cells, the magnocellular cells, and that goes to the superior colliculus, and that in turn generates eye movements and your visual grasp reflex. Now, in addition, you have another pathway going from your early visual areas to your parietal eye field. That was one of those areas that we talked about just a moment ago. And from there to the prefrontal cortex, a structure that's very similar to the superior colliculus called the frontal eye field, and then down to the superior colliculus, and again the structures that generate eye movement commands. So this area is, if I wanted to look at one of you, and I remember that one of you is sitting to my right, and I can quickly point my eye to that person because I remember you sitting in that spot. And to do that, of course, it needs memory, which comes from your prefrontal cortex, and attention, which comes from your parietal eye field. And these things all work together then to fire off the frontal eye field and again generate a voluntary saccade. Whereas the direct pathway was sort of an involuntary saccade when something unexpected happened, some louds, some hand being raised, or a light flashing, or just a sound being made. Now, this information to the superior colliculus fires off a large number of neurons because they have big receptive fields. But it's good to think of this activity as being broad, but also peaking at a hill. And this is the location of the maximum activity that gives you a good estimate of where the location of that object is. So thinking of these things as hills of activity is probably important in figuring out how it is that saccades are very accurate to the objects that you see. Now, one of the things about this is that when an object appears in your periphery, you then make a saccade to it. That then moves that activity to the fovea. So you see something in the periphery, you make the saccade to it. That means you point your fovea at it. That means now the activity will be appearing in the foveal region of your superior colliculus. The other thing that's important to consider is that in order to make this saccade, activity in the frontal eye fields and in the foveal region actually inhibits saccade. So if information is coming from this part of the superior colliculus, it generates activity which suppresses saccade. So it initiates fixation. Fixation is an active process. To stare at something is an active process. You're inhibiting some neuron from firing and producing a movement of the eye. So then when something appears in your periphery, the first thing you have to do is remove activity from your foveal region. And then this activity in your periphery generates a saccade and then moves the activity back to the fovea again. And activity again suppresses, in the fovea suppresses another saccade until something else appears that you want to move to. Now, the map we were looking at, the important thing to remember is that one half was on one side of the brain, and the other half was on the other side, as we'll see in a moment. But this map that in the superior colliculus is similar to the maps that we'll be talking about all along this stream. So we have information coming from the retina, from V1, and the types of maps you'll be getting changes somewhat. In V1, we saw that the fovea had a very large representation because the axons from your optic nerve favored the fovea. There were many axons coming from the foveal region. So this then got itself a large representation, and the periphery then a smaller representation. These areas here care about where objects are in your periphery. So they have a large peripheral representation and a small foveal representation. The other thing here is if you, either because an object appears here or you stimulate it, if an object appears here, this activates neurons here, which turns the eye towards the object. If instead this object appears here, then activity occurs here and turns the eye towards the object again. If both these hills became activated by, let's say, stimulation, then the commands would cancel themselves and no movement will occur. And as I mentioned just a minute ago, all these structures are on both sides of the brain. So even though this looks like one single map, one half of it is in one cortex, the other half of it is in the other cortex. Like the representation of V1. One half of V1 was in one half of the representation was in the other. It's the contralateral that's being represented. So everything on the right is represented here on the left. Okay, let's look at what the activity of these structures is for 4 simple tasks. So the first one is this A flashes on over here, and you're asked to maintain fixation. So keep looking straight ahead and not focus your attention on letter A, ignore letter A. What happens what would happen in your cortex? Well, the only activity that would happen is that a stimulus would appear in the peripheral part of V1. Okay? Because you're not attending to this A, none of these structures would be activated except the frontal eye field, which has an activity in its foveal area. Because that's suppressing saccades. Now, if you had another task, which was when A appears, look to A, okay, you'd get activity in the following areas. First, we'll again have activity in V1, but because you're tending to A, you'd have activity in the parietal eye field, and because you're making a saccade to A, you'd have activity in the frontal eye field. And then to the superior colliculus, and then making a movement to A. Now, let's say you have had another task. You have stimuli appearing at A and B, but you have to make a saccade only to B. So your task is only to B. What activity would you see in these areas? Would you see activity in the right V1? I see some nods. Okay, hit the table if you think it'll be in the left V1. What about the right PF? Nope. What about the left PF? Good. Right frontal eye field? Left frontal eye field. I pressed too early. Anticipating what you'd say. And you're right. That's exactly where activity would appear. And then you'd make a saccade to B. Okay. So what happens if you're asked, again, A appears, what you're told not to make a saccade to it, but to reach towards it or to point towards it. So what happens here is you're asked to tend to A because you don't know where A might be, but when it appears, you point to it. So you have activity not only in the visual cortex, but you have activity in the parietal cortex, but this time in the parietal arm field, as well as the parietal eye field. Okay, the parietal eye field, it fires not only for eye movements, but for anything that you attend to. Okay, so you have activity there, but as well you have activity in your parietal arm field. These two regions here. And because you have to maintain fixation, not look at A, you have activity in the foveal region of your frontal eye field. Okay. So in summary then, the retina and the visual cortex is activated whenever there's only a visual stimulus, no action required. The parietal eye field fires when there's visual stimulus and attention, and one attention, in fact, that it's being selected as a target for movement. That movement can be either an eye movement or it can be an arm movement. And finally, the parietal eye field and the colliculus get activated anytime you have a stimulus, you have a tension, but also an eye movement. If instead you need an arm movement, then it's the parietal arm field that gets activated. Okay. Now, let's suppose a target appears only briefly, and you're asked to attend to it, but not look at it. So again, you maintain fixation, but because you're asked to attend to it, you get activity in the parietal eye field, but you also get activity in the prefrontal cortex here. Okay? And that holds this object in short-term memory, your working memory. Now, if, to make things even more complicated, you're shown two objects, A and B, and you're asked to saccade first to A and then to B, but not when you could see them, but when they're gone. Okay, so you're saccading not to the actual visual input of A and B because it happens too quickly, but where you remembered seeing them. So how do you do that? Well, you can see that initially you get activity appearing in your visual cortex in sequence, and then in your it appears in your parietal and your prefrontal areas, and it maintains there in a tonic way. So this one just appears in the visual cortex briefly, whereas here and here it appears in working memory for some persistent time. Now, let's see what happens after you make a saccade to the first object, the letter A. Okay. When you make a saccade to the first object, it isn't there. You're just moving to its memory. So you got this location stored in your prefrontal cortex as to where A was. You make a saccade to it, activity shifts in your prefrontal cortex after the saccade. and over here in the parietal eye field. And it appears now, A appears now in your folial. That is where you would be looking at A, were it visible. Okay. Now, the question is, what moves this? What shifts this thing from here to here? Well, it's our old friend corolary discharge, and that corolary discharge originates, we'll see, in the superior colliculus, and is sent to the frontal eye fields and the prefrontal cortex and the parietal eye fields. So this corolary discharge shifts A and B, and it shifts A to where it would be where visible, and B to where it would be if you were to make now a saccade to it. Okay? Now, in everyday life, you make your saccade. I make saccades to people in this room, and I sense they don't move, even though my eyes sweep across the room from here to here. There's a lot of movement on my retina, and yet I don't sense that anyone's moving in the classroom. And how is that so? Well, what we see in our visual cortex moves and appears where our corolary discharge shifts it to be. Okay, so A appears in your parietal eye fields where the vision where A actually moves to because of your eye movement. Okay, so your eye moves, it generates, it has a corolary discharge to it, and now appears where the actual A is. And as in the lecture 3, we know the lion hasn't moved because things are where they would be had the eye moved. Okay, the last thing I'd like to talk to you about is something called overt and covert shifts of attention. Overt shifts of attention are the things we just talked about, saccades, making actual eye movements. They're called overt because you can see the person, where the person is paying attention. Covert ones are where in your mind you're shifting your attention. You're fixating something, but you're thinking, well, maybe over here there might be possible objects I should saccade to. So you can see here, covert shift here, covert shift here, covert shift here. So you're studying possibilities of where Waldo might be, or something of interest might be. And because these covert shifts, you can make them a lot quicker. They're useful in terms of determining where you would make an actual eye movement to. And then you land there and you say, oh, that is where that activity is. So these covert shifts, they enhance the activity. So the covert shifts produce activity both in the frontal eye field and the parietal eye field, and the activity enhances activity all the way into V1, which increases the contrast in V1 and allows the visual system to examine this part of the visual field and say, hey, is that Waldo or not? Should I look there? And then if it is Waldo, you make a saccade to Waldo. And you say, yep, that's where he is. So, how all this happens is not clearly understood. So, what we have to do, remember, you have to, for the overt shifts of attention, you have to maintain activity in the foveal region in the frontal eye fields. But at the same time, activity does happen in the peripheral areas of the frontal eye fields that don't initiate saccades, so that did initiate the covert shifts of saccades, but yet they don't generate a saccade. So in the next session, we'll look at the somatosensory area in more detail. And we'll see that it's a strip just along here in the somatosensory, in front of the central sulcus, that codes your body. And there's one strip that codes all the touch information coming from your body, and another strip just in front of it which codes the activity coming from the muscles and joints underneath your skin and telling you where all these body parts are located. So whether your thumb is extended and your index finger is bent, so all that information comes from things inside the thumb, in the joints and the muscles. And all this helps you generate maps of peripersonal space. And these maps are normally what we can sort of reach for, but it can be extended. If we take a pointer, the area space that this represents gets larger. If we sit in the car, this area space becomes larger still, and we know exactly where the edges of your car are, and if you're good at it, you can then park your car very accurately in a tight situation. Okay, that's it.